This presentation will describe how the IROA protocol can be used to measure the metabolic response of an organism to a physiological change or genetic alteration. The key advantage of the IROA protocol is the creation of an internal control standard for every experimental metabolite so that a direct, precise measurement and the identification of each compound can be made. Our focus at IROA Technologies is at the biochemical level, closest to the phenotype. Generally, the size of the metabolome is smaller compared with that of the genome or proteome, making the biochemical content easier to measure and understand physiology. Even with advances made in analytical mass spectrometry, there are numerous bottlenecks to overcome in metabolic profiling. The IROA tools were specifically developed to address these bottlenecks. There are two components to the IROA methods. The first involves using specifically designed cell culture media to label the complete biochemical content of a living organism with an easily detectable molecular signature. The second component is the software algorithms called Cluster Finder that can easily find and verify these signatures, and even more importantly, remove all artifactual peaks which often lead to false discoveries and inaccurate data interpretation. Almost all living things contain two isotopes of carbon, carbon-12 and carbon-13. Generally, the natural abundance of carbon is approximately 98.9% C12 and 1.1% C13. Consider the molecule arginine shown here with a formula of C6H14N4O2. This molecule has 6 carbons, and given the natural abundance of carbon, each of the 6 carbons has a probability of 98.9% that it is a carbon-12 and a 1.1 probability that it is a C13. That means in a mass spectrometer, arginine will have a base peak at 175.119 AMU and a very small peak called an M+1 at 176.122 that is approximately 6% of the base peak's height. The small size of this M+1 peak makes it very difficult to find and generally useless for data interpretation. If the percentage of C13 is increased to 5%, then the M+1 is significantly larger, now 32% of the base peak's height, and can be easily detected. Furthermore, if the C13 level is increased to 95% C13, the peak arrangement is now dramatically changed, and the base peak now shows at 181 that is entirely C13 and has not an M+1, but rather an M-1 peak at 180. If we put these peaks all together, we can see that these three peak classes, natural abundance, 5%, and 95%, can easily be distinguished. They are all mathematically calculable as well. That means that the Cluster Finder software algorithms can easily go through and identify if a peak contains natural abundance, 5%, or 95% carbon. Furthermore, the 5% and 95% peaks for a given compound are naturally paired with one another. That provides a tremendous amount of additional information. As seen here, the fact that this molecule contains 6 carbons may be determined by the distance between the two base peaks, and the relative height of the M+1 and M-1 provide confirmation of this fact. This triply redundant confirmation provides a high level of accuracy. How do we generate and use these unambiguous chemical signatures for metabolomics? Like SILAC, the IROA method utilizes metabolic labeling, and cells are grown on isotopically defined media, but it is based on labeling compounds with carbon sources C13 enriched at either 5% or 95%, instead of labeling with C13 enriched at levels greater than 98% to create specific patterns. Unlike SILAC, the IROA method labels all biological compounds and not just specific compounds. Any cell grown on such a media will effectively show a complete labeling of its entire biochemical content if it is allowed to grow for at least 5 cell divisions. All biological compounds will carry a distinct molecular signature, and these molecules can be distinguished from each sample set. Experimental, grown in 5% C13 media, or control, grown in 95% C13 media, as they have differing masses. Every molecule from cells grown in the 5% media will demonstrate a strong M+1 peak, while every molecule from cells grown in the 95% media will demonstrate a strong M-1 peak. After labeling, the 5% cells may be treated with a stressor, for example a drug, and because of these distinct patterns, they may be directly compared with the 95% control cells in a single pooled analysis. Using a pooled sample dramatically removes technical variability. During the data analysis, the cluster finder software can identify and remove all unsigned molecules, which can be classified as noise or artifacts, as they will not have either a strong M+1 or M-1. The removal of artifacts, usually the largest portion of the data set, is generally not possible in current metabolomics protocols, where the high number of observations increases the false discovery rate. Another important consideration when using the IROA protocol is that suppression is also removed as a source of variance or error, as identical molecules labeled with either 5% or 95% C13 during mass spec analysis experience approximately the same ionization efficiency and suppression. Because samples are isotopically labeled, the IROA technology enables compound identification. Using the knowledge of both accurate mass and the number of carbons, a molecular formula for each biochemical compound can be calculated and the compound readily identified. Generally, these compounds would remain unnamed. Please see the video on compound identification to learn more. There's another way to utilize the IROA protocol that can be used when it's not possible to label experimental samples, such as tissue biopsies, field-grown plants, or large bioprocess fermentations. Please see the phenotypic experiment video for further information, or contact us at info@iroatech.com. Thank you.